Yeah,
Oh,
see, Citrino pallida is pretty yellow. I was thinking of that one, but it's uniformly yellow and it's a western species, mountainous.
and the best way
So
to see the good photos and do it right is to
it
go to
could be a faded lilacifolia. They have
probably
a yellow cap,
see now I was hoping it was something super weird and like new, and now I'm just like, oh, it's just gonna be Chromosa.
but that's cool.
Just a chromosera.
There's not a lot of records in Wisconsin. I think that'd be great if it was
That's
one.
true.
And
And
it's a
the DNA
newly
will
revised species too. So it actually, now that things were all in cyanophylla and pulled out, it's more helpful to have things go in their with their regional species name. So I think it's
mhm.
perfect timing.
Well, with some beautiful Waxies already being shown off, I will say we've hit 7. So we've started recording the meeting, and I can also introduce Lauren with her bio for anyone who doesn't already know Lauren. So, Lauren Ray is a mycologist and nature educator newly based in the Midwest. She lives in La Crosse, Wisconsin, where she is pursuing a master's degree in biology at the University of Wisconsin-La Crosse. Her research focuses on the systematics of the fungal family Hygrophoraceae, known as the Waxy caps. She holds a Bachelor of Science from the Evergreen State College in Olympia, Washington, where
Woo.
she studied ecology and cryptogamic organisms, including fungi, lichens, and bryophytes. She is passionate about community science and fostering greater awareness and understanding of fungal diversity. So that's who Lauren is, and she's here to present on Waxy Caps.
Thank you so much, Mariah, and thank you everyone at WMS for having me. I've been wanting to do this talk for a while, and I'm glad there was an opportunity to pop in and give it to you guys. Like I was saying earlier, I gave this talk to NAMA first, and so it's pretty much like going to cover it from a wide perspective. We're going to talk a lot about like the background of Hygrophoraceae, who's in it, what do they look like, what's their ecology and stuff, and hopefully it can be informative for Midwestern diversity as well, just a bit, but. Yeah, I'm gonna share my screen and let's see. I haven't done this in a while. I haven't done a Zoom talk in a long time. Okay. It's probably, how's that?
Good.
Okay, I actually need to make the little Zoom column small so that it's not in my way. So just give me a moment. Okay. Perfect. Okay. That's good. Yes.
Yeah. Sorry. Yeah, looks
Okay,
good.
cool. Thank you. Okay, perfect. Well, I'm excited to talk to you guys today about waxy caps, and I call this talk Beauty, Mystery, and Discovery within Hygrophoraceae, which is the waxy cap family. And I'm Lauren Ray. Let's see if this will let me progress. Yeah, so an overview of the presentation, we'll talk a bit about the history of the family, some distribution, ecology, community science projects and findings that we've had, some discoveries we've made, and just a bit about identification as well. You already heard about me, and I've been lucky to meet a lot of you. But I guess in case you didn't know, I grew up in New York and got into mycology right before I left the state to move to Washington and finish my degree over there at Evergreen, which is really well known for having studies in these cryptogamic organisms like we just heard that fun word. So this hidden cryptic organisms, they have this hidden lifestyle and hidden reproduction specifically. And so that includes the fungi and mosses and such. So I was really lucky to get out there. That ties into to how I got involved in this project that I'll be talking about. And I'm currently living in La Crosse, Wisconsin, and getting my master's in science here at the University of Wisconsin in Dr. Arthur Group's lab. And with Mariah, woo! And so, yeah, I also like slime molds, other tiny little things. I love small mushrooms. It's kind of fun. This picture shows some Hygroscope fulvescens that they're already a larger wax cap, but these were particularly big ones. But I tend to be inclined towards more of the really small and obscure things. And I'm really just huge advocate and participant in, I guess, education and science, specifically with mycology, because there's so much that we need to learn and share within this field. So I'm really lucky for all those opportunities I get to share my passion. Okay, there's so many Zoom components going on, but it'll be okay. All right, the history of Hygrophoraceae. So the type genus was Hygrophoraceae, and this was in 1836 by Magnus Fries. And so you'll hear Friesian taxonomy a lot because he named a bunch of mushrooms, and we kind of follow his taxonomic style. Um, but the family was formally created in 1907 by Johannes Lotzi, and he wanted to kind of group together some of these mushrooms that had really thick and waxy gills with white spores. And so they're called waxy caps. Part of this is specifically referring to these waxy gills. And people ask me about that sometimes. And so if you haven't tried it yet, if you find a wax cap, pull out one of the gills with your fingers and crush it. And you'll actually feel that it feels like candle wax. So that's where that name comes from. They're also called wax caps in the UK a lot. You'll see that. And they weren't really fully incorporated into mycological taxonomy until Rolf Singer's revision of the Archaeles in 1951. And that's still when fungi were plants. So, taxonomy's come a long way since then. We have a pretty good concept for the family now, but it's always evolving. We have currently 28 genera in the family and over 600 species worldwide. This number really is a low estimate. Like, this is the currently described species, but there's going to be a lot more that we still haven't encountered and that we are currently discovering through the sequencing efforts and community science projects. So there's a really diverse morphology and ecology within this family. There are more forms and shapes and colors than you've even probably heard. So they're not all perfectly bright rainbow mushrooms, and they're not even all gilled. So we'll talk more about that and some of their ecology. They can really vary in their roles. I love this illustration here by Beatrix Potter, who is known as being an author and illustrator, but was also a mycologist and did incredible experiments and illustrations and documentations of mushrooms. And she even included the spores in this illustration. Such a accurate depiction of this Hygrophoraceae coccinea. And for the etymology, hygro referring to water and forest bearing. So it's water bearing. A lot of them are super slimy. So that helps to remember that. Here are some of the North American genera. There's a lot of them. A lot of the names can be like strange combinations of each other too. And we won't go through all of them, but I highlighted here these three in this blue box: Gleophorus or Gleophorus, Hygrophoraceae, and Hygrophoraceae, because these are the genera that most people think of when they think about waxy caps. Particularly Hygrophoraceae, because those are those really super bright, iconic mushrooms. Gleophorus too can be pretty brightly colored. Hygrophoraceae are more montane, though they are widespread in temperate regions, and they're usually less colorful and a little bit less typical of like what you think of with. They have waxy gills too, but the Hygrophoraceae really shine in people's minds. And then here is a screen cap that is from my original version of this talk from April 2022 from iNaturalist, showing the observations at the time for the entire family throughout the world. So there was 86,087 observations. And I was so excited to see what the current update was. And there we go. Oh, I didn't even put the total. It's 257K around or something. So there was 189,000 observations added or so in the last 3.5 years. So that is really exciting. You can see Greenland's got more observations. If you look up there in the north, in the Arctic areas, there's some observations throughout the northern parts of Russia as well. And just throughout the world, there's a lot more people getting out and documenting these mushrooms. So that's really exciting. And then this is a screen cap of the taxonomy in iNaturalist. just to kind of further show where we are in the fungal kingdom. So we're in the Basidiomycota, which is a lot of the mushrooms we talk about, and then in the order Agaricales, which I've starred here. So it's the common guild mushrooms. Not every guild mushroom is in this order, but this is just kind of generally speaking. And then there's the suborder Hygrophoraceae, which encompasses Hygrophoraceae, their subfamilies, and then there's sections inside the genera, and there's subspecies. There's all kinds of breakdowns, and it's actually being pretty hotly debated right now if these subfamilies should be their own families or not. There was even a paper that came out that was trying to do that, but we'll see what sticks because there's a lot of different things to consider with taxonomy here. Okay, so some more fun stuff here. Ecology. Talk about some of the major ecological roles within this family. So the Hygrophorus, these are ectomycorrhizal. And so this means they're mutualistic with trees. So they have a mutually positive exchange with the roots of trees, just like a lot of the russulas, Lactarius, Amanitas. So they will exchange water and minerals for sugar from their host tree. And so this is, I believe, the only ectomycorrhizal genus in the family, but this is the one that's most well known. And well, yeah. And so here's a couple of different species. Speciosus has been found couple of times in Wisconsin. Actually, I was able to see some records for this. This is associated with larch. So we have the tamarack here, and there were some observations of that species, but it's common out in the mountains in the west. And then some of these other species, a lot of them are associated with conifers too. So there's pine associates. Some of them can even be parasitic there. So they're like, oh, we know they're ectomycorrhizal with the pines, but there was a study that showed that they were actually even causing some stress and kind of overly pulling their share of sugars from their host tree. So these are some of the bigger fruiting bodies within the family, and you can see those really thick, waxy gills, and a lot of them, they tend to have more pale coloring. Some of them can be brightly colored like speciosus, but there's a lot of browns and grays. There's some pretty pinks and reds as well. But yeah, these ones would be, I would say, some of those that might be harder to pin down as a wax cap if you were not used to encountering it. And then there's saprobes as well. So Pseudo-Armillariella, that's a fun one. It has been found in Wisconsin as well. And I guess it does look like honey mushrooms, I'd say. So there's armillaria is the honey mushroom, so it's being called Pseudo-Armillariella. And it has those little hairs on the cap, this yellow coloration, clustered growth on wood. So you could see how it might resemble a honey mushroom. And that's a pretty interesting species. I've been lucky to see that one. Curious if anyone in the club has seen it. But Cremosa, we talked a bit about Cremosa too. This is a beautiful wood decaying saprobe in Hygrophoraceae that have a lot of bright colors, the yellows, pinks, purples. And there was a recent paper that I was lucky to be included in that kind of further elucidated the North American species, and I'll talk more about that. Chrysomphalina is not present in the Midwest, but it's very common in the West Coast. And there are a couple different species that are very brightly colored and strongly clustering on wood. Arenea, not all Areneas are saprobes. I'll show you some that are growing on moss, but this is one that Epichysium, I don't even know if I've ever said that out loud. That's a common species on wood that you can find in this region. And those tend to have darker coloration. So biotrophic, what does this mean, biotrophic? Bio is, alive, and trophic is they're referring to their eating and their food. And so hygroscopies for a long time were thought to be saprobic. Some of them can be saprobic at times, but a lot of them are actually getting their nutrients from living organisms. They're not necessarily in a mutualistic exchange. They're not necessarily parasitizing and harming it. They might just kind of partition off a little bit of living tissue, say from a moss, or maybe within some grasses. We'll talk more about the grass situation. There's something interesting there. But they can be found growing on wood, in which case they can kind of opt for a saprobic life. But a lot of the times you'll find them in mossy banks that have been disturbed. And so it's more so that they're actually eating from that moss than they are eating dead material within the soil. So that's part of why you find them with these associations. Oh, yeah, and these are some really nice examples of moss dwellers, or species that are always found with moss. So sometimes they can go, some species can go either way. You can find them in moss or just in some bare soil or maybe in some grass, but these are species that are always going to be occurring with mosses. And so Arena chlorocyania is fun. They look bigger in this photo than they actually are, but the moss that's actually juniper haircat moss. It looks so big, it almost looks like grass in that photo, but it's actually a pretty big moss. Um, and it's such a gorgeous blue mushroom. This is one that I did not check if it's been found here. Does anyone know if it's found in this area? Give it a second. Chlorocyania. It's a gorgeous one.
No.
No, it hasn't. Yeah, it likes this moss, but it also can grow in like the sand dunes as well, with the moss there as well. So potentially they haven't been found yet. Maybe they are here. You need to go check. Arenia rediruga, that's a really strange Arenia. These three are in the same genus here. It's the outer ones, or the first two on the top and the first one on the bottom. And you could see how diverse that one genus is with this strange morphology on rediruga, this kind of like cephaloid cup wrinkled mushroom. That's the whole mushroom there. That's the fertile surface kind of showing underneath this wrinkled part. And then you could see the top of the cap, but quite a strange one, no stipe. Yeah. It's a cool species. And then Sphagnicola with sphagnum, and then Cuphophyllus cinerelis is a gorgeous bluish gray species that is also associated with sphagnum in bogs. Okay, so there are also lichens in this group, which a lot of people, you'll hear like kind of general descriptions of lichens, and people really strongly associate them with the Ascomycota because majority of them are in the Ascomycota. It's about 99% of lichens are in the Ascomycota, but it's not true that they all are. We have a bunch of lichens, not just waxy cap lichens, that are basidiomycetes. And so we have a couple of different genera in the Hygrophoraceae that are lichenized. These are a more common genus, Lichenomphalia. In temperate regions, you'll see these mushrooms here. They were Lichenomphalia. I learned it as umbellifera. It's recently moved to Ericitorium, and this one is common. This grows in. I haven't seen it here, but do you guys find it in Wisconsin? My live consultation.
I think I've seen observations on that. I've never seen it myself.
Okay. Yeah, it's interesting because it's funny too how sometimes when something is super duper common in one region, it can and then you don't see it when you're out and about in another area. You might think, oh well, it must not be here, but it's just possible that it's more rarely encountered. And so, like in Washington, this mushroom is just all over the place. The dead stumps are covered in this green film, so you could see these green bubbles surrounding these young mushrooms in the first photo. And they're actually these like combinations of fungal hyphae wrapped around these algae and creating these kind of green balls and green films all over the wood. And then you get these gorgeous whitish, creamy mushrooms with the decurrent gills and the scalloped margin. They're just gorgeous. I also included two species from different regions. Lichenomphalia altoandina is in the Andes, and it's one that I'd love to see up in the Atacama Desert in Chile. They're in the salt crust, so the fungus is actually able to find its algal partner and grow in this really extreme dry, salty habitat and create these little bright orange mushrooms. And I think that's just absolutely fantastic and interesting. So if anyone gets down there and finds it, please let me know. There's only a few observations of it. And then Lichenomphalia hudsoniana is a more northern Arctic and subarctic species that has these more squamulose thallus, so the vegetative the part that has both the fungus and the algae are going to make this leafy green structure down at the bottom, and then you'll have just the fungus making a fruiting body, making basidiospores. So it likes colder climates, that species. These are some bizarre and very, very enchanting basidiolichens, I'd say. They look like turkey tail to me, like the blue turkey tails, but that's actually the full, that's a thallus again. So this is the full lichen with its fungus, with its cyanobacterial partner in this case. So the previous one we saw was a green algal associate. These are associated with cyanobacteria. And so that percentage was giving us like 1% of lichens are basidiolichens, and then or less than 1%, and then less than 10% are cyanobacterial lichens. So a majority of them have green algae. So this is a super rare and unique combination of symbiosis. And you can see on the Cora, I believe all three of these genera will do this, but they make these cephaloid basidiocups. So what that means is it's just kind of sometimes cup or disc shaped, kind of maybe a little flat or sometimes even rounded. So that white, those little doublies in the on the shelfy structure is actually the fungal fruiting body where the spores are made. And they're in South America and a bit in the tropics. They can be found in more tropical wet habitats, but also up in the mountains as well. And these genera have been split apart. So they were originally, I believe, Dictyonema or Cora, and then separated out. But something that's interesting, I know for sure Dictyonema, this is the case. I'm not sure about the others. There is some very odd chemistry. There are actually multiple psychoactive components to these lichens. They have psilocybin, just like psilocybe or the common magic mushrooms, but they also can produce DMT, a different psychoactive compound. And so I sometimes avoid telling people about these because I don't want everyone to book a ticket and go like into the jungle and try to find these lichens and rip them off the trees. But if you do encounter them, leave them be, document them. Some of them have even thought to have been extirpated from Florida, potentially previously living in North America in that subtropical or like that kind of tropical corridor, but then with development pushed out. So I just, be respectful. If you do end up going down there, I'm sure if you're going down to look for mushrooms, you'll be respectful. But yeah, this is just there's a interesting history too. Different shamans in the area have used them to even like curse one another. So it's kind of been used to rival and maybe poison or curse other shamans. This other basidio-lichen, acantholichen, this is another member of the Hygrophoraceae. I kind of thought that the middle photo looked like a sour gummy, maybe like a Halloween goth gummy. But there are these super interesting leafy structures actually growing on liverworts, you can see, and mosses. And so this genus there's not a lot of species in this genus. And yeah, they are also not found in temperate areas, but I would like to see them one day. Okay, so wax cap grasslands. This is kind of an ecological phenomenon and also an important conservation metric and habitat that is used in the UK. And so in that region, there are these large swaths of grassy areas. They're kind of like prairies, but they're slightly different. There's a lot of these annual plants, and there's actually pretty low plant diversity, but they are a native habitat. So it's low plant diversity, also very low nutrient, but there's very high fungal diversity in these habitats. And some of the highest diversity that's been documented is within this grouping of 4 different genera that we abbreviate as CHEG. That's the second bullet there. Clavariaceae, so some of the clubs, Hygrophoraceae, our star, Enthylomatacea, so the paint gills, and then Geoglossaceae, so an ASCO, the earth tongues. And then the D is for Dermaloma, a genus in Trichomataceae that sometimes gets included. But yeah, these habitats are very sensitive. They tend to be converted into farmland and grazed. If the grass is over, like they believe like that the natural habitat would involve partial grazing as different ungulates would move through the area, but then have chance to kind of for the grasses to grow big and tall again. But that low nutrient component is important too. This is a lot of times with the agriculture, there's heavy fertilizer that gets added, and that will throw off the balance and push out this diversity. And so for ecologists who've been trying to understand how much of this habitat is intact, they'll actually go and count how many species are present within these 4 families. I love in the mycology graduate lounge at UWL, there was a calendar somebody pulled out. I don't know if you probably can't see it with my camera, but I love that there's an example of a waxy cap and a club together. So it just made me think of our beloved Chegg. But yeah, so the habitat is super, super diminished. There's very few left. There are researchers who go out and document these, but they actually wanted to offer the public the opportunity to get involved and see if they could find maybe some of these remnant habitats. And that's where this wax cap with funny lettering is part of that. So this is an app that was made, a citizen science app that was made to help people get out, walk around their neighborhoods, their areas, and see if they could find any of these mushrooms that might be an indicator for this habitat. And through this project, there were over 80 sites added to the researchers' studies. And so it's just kind of a cool example of how community and citizen science can push research forward. Something that was learned from these habitats is this endophytic lifestyle. So this is, like I was saying, a lot of Hygrophoraceae and similar genera were thought to be saprobes and just decaying like grass roots. But there were some experiments that were done in Europe and then also some replicated in North America that show that the multiple different members of the Hygrophoraceae, a lot of them tend to be the Hygrophoraceae, but also Kupophyllus, will live inside of the living plant tissue and actually be transmitted as the plant gives its seeds off. So they're vertically transferred. They're from the parent plant. They can actually be passed on and like kind of be dormant within the seed. And as that new seed germinates, the fungus can also grow along with it, and it can kind of slurp up nutrients as the plant is growing. But they're not only in one part of the plant, they can be in the seeds. And then part of what this crazy diagram is showing is that despite sterilizing the plant seeds and then trying to inoculate them with different species, they actually ended up getting different species than the ones that they were purposefully trying to inoculate. So it was kind of the experiment gone wrong, but also showing them that there's this persistent diversity living within these seeds. And you can have other species living in the roots, in the shoots, and leaves, and in the seeds. So you can have up to 3 or 4 different species within one plant. And this was done on Plantago lanceolata, one of the plantain plants. So this is fascinating, and it's been documented in more than just Plantago, and we know that this is occurring across different habitats. And yeah, so it's curious to, oh, well, I'll show you that. I was gonna say it's curious about the difference in habitat between North America and Europe, because like we're saying, in Europe, there's so many of them occurring in these grassland habitats, but it's more typical that you'll encounter them in forests in North America. And we have not yet seen that they're endophytic with these trees, but they do come up sometimes in soil samples and some root samples, but we don't think that they're living the same way with these trees. So it's unclear at the time. I was excited to get these photos today from my good friend and our club member here at UWL, Jamie Yeager. And the 2 pictures on the right are actually from today, that they found this mushroom today. This is a parrot wax cap found in a prairie over in Prairie Moon Nursery in Winona, Minnesota. And so they have this prairie that they're actively growing plants in and selling different plants. It's a little bit different than a traditional prairie, but it's got a lot of the similar plant diversity overlap. And they actually found this parrot in this on-site prairie, which is super fascinating, not a common occurrence in North America. There have been waxys documented in grassy areas, but like I said, majority of the time it's in the forest. So keep your eyes out in prairies and grassy fields to see if you can document some of these wax caps. So this is a lot of times this is the parrot wax cap. It's usually more green, but there's a lot of range in the color, and you can see there's a little bit of green on the bottom photo, but a really beautiful bunch and really interesting to see in this habitat. And then they have another observation of a Hygrophoraceae from a different time in the same prairie. So keep your eyes peeled. We have a lot to learn about the ecology. So I also want to talk about the Hygrophoraceae of the Lower Puget Sound, which is a wonderful project that I got involved with as soon as I moved over to Washington for my undergrad studies. And it was started in 2017, and it was part of the North American Microflora Project. It is now the Fungal Diversity Survey, or FUNDUS. And basically, if you haven't heard of it, it was a really cool initiative where they put this offer out to the public, and they said there's any group of mushroom that you're curious about, want to learn more about, you can register a project and you can pick an area to study. So it could be like a specific small city park, or it could be as big as like the entire West Coast, or maybe the state of Oregon or something like that. And you could narrow in and choose a specific group of mushrooms, like Steve did with the Hygrophoraceae, or you could just say, I want to document the whole fungal diversity, everything I find from this park or so. And so Steve is an incredible amateur community mycologist. He's a retired carpenter with no scientific training, and through the South Sound Mushroom Club, he set this project up and encouraged members to go out and make collections and preserve them, voucher them, dehydrate them so that we could study them further. And he applied to get free sequencing. At the time, it was pretty tricky to get sequencing as a community member. A lot of times sequencing, it was only available to academics, or if you wanted to do it as a non-academic, you had to pay a ton of money. It was like $30, $40 a sample sometimes. And we even paid that when we ran grants at times. But luckily, we received multiple grants and were able to get tons of specimens sequenced with a lot of interesting results. We also got partnered up with Dr. Jean Lodge, who is one of the world experts on this family, and she was looking at our sequence data and helping us figure out what was what, and kind of there was a lot we didn't know. We still, the more we find, the less we know in some cases with these things, and so she's just kind of helped us navigate this and figure out what we've been finding. And so, yeah, it's been a lot of collaborations between all kinds of the club members, students at the universities, but also just it spread kind of like wildfire. People just got really excited about this project. They wanted to get out and find these beautiful mushrooms. And so they were sending us collections from all over the West Coast, and we were able to include those. Here's kind of some pictures showing the process. It's not a total linear process. I kind of showed the first part as a little bit linear, where we have someone getting out, as our friend Sophie, getting out, finding a mushroom, photographing it in the field, trying your best to get photos of what it's growing on and the size, shape, color while it's fresh, and then keeping them separate in tackle boxes. Here's an incredible, colorful array of wax caps from one of our days out on the college campus. And then a dehydrator tray showing how we can preserve these mushrooms. We want to get them crispy and save them so that we can pull tissue for DNA and also do other things. Like down here, we see some microscopy. There's Jamie there. We could do some DNA sequencing. There's Mariah doing some extractions, and also sit with keys and other resources and documents and see if we have any idea what we're finding, what's available in the literature. But yeah, there's a lot of ways you can do this. Sometimes one person does part of the process and then other people pick it up. Maybe someone finds the mushroom and then another person takes it home and saves it, and then someone else does the sequencing, and maybe someone else does the micro. And so it's very collaborative, and if these end up getting included in manuscripts for scientific publications, which some of them are, you'll see that lots of different people contribute to different parts of this process. Here's this kind of the cover of our iNaturalist page here. You could check it out. It's available for anyone to look at. And it has grown a lot since 2017. We have lots of different, it says 10 observers. There's another project that has other collections as well. And so that's not been only 10 people who've contributed. We've probably had a couple dozen at this point. And so I just wanted to talk about some of the findings that we've made. So this is a mushroom that is a red listed species, Hygrophoraceae flamifolia. It was listed as a Gleophoras at some times, but it's we believe to be a Hygrophoraceae, and it is a threatened species. And it is included in the West Coast Rare Challenge list for the Fungal Diversity Survey. So it was included in this list of species that they wanted people to get out and make more observations of. And it was only thought to be found in as far north as the Redwoods in Northern California. And Sadie Hickey, our club member and grad student here too at UWL, found it on our campus. And we were so excited to see this mushroom. We had no idea we'd be able to find it this far north. And so it was the first record. You can see the lower observations in California and that 700-mile range extension from this observation. And then I was able to find it and photograph it the next year. So those are my photos from my collection. It's got these golden, bright, beautiful gills, flavofolia, golden like foliage or gills in this case, and sometimes this like whitish, sometimes even purplish cap center. Super, super viscid and slimy mushroom. That's why it was thought to be a Gleophoras. So that was a big deal for us. This is one that I found that was also a range extension from a lot of these species were just like the western waxy cap species were thought to hit as far north as Northern California. So when we were getting out there on our college campus, there was so much diversity, and we ended up making a bunch of these records and extending our concept of where these mushroom species were thought to grow. So this is Latissima. I broke the one in the middle. It looks like it has a veil, but it doesn't. This is actually a very large species and has this very straight yellow stipe and this bright, like orangey red cap. So I was lucky to be able to write up a little species summary for the Burke Herbarium in Seattle so they could add it to their Washington, a species that occur in Washington state. And this was right on the side of the trail, as were these Hygrophoraceae virescens, the lime green waxy cap. And they had, I believe, been previously documented in Washington, but not collected. I think there was kind of more word that they had occurred in the more far north. They were more well known from California, but they had this whole strange history of irregular fruiting, where in David Aurora's book, Mushrooms Demystified, he talks about how he would go out to the same areas for collecting mushrooms. He'd go out all the time throughout the year, and he encountered this mushroom species, and then he didn't see it again for like another 7 years. And then as soon as it popped out, it fruited all throughout the coast at the same time. And people were finding all throughout California, and then not again for a bunch of other years. And so we were able to find this mushroom in Washington, document it, collect it, get it sequenced, and then we actually kept finding it multiple years in a row, which seemed to be unusual. So that was part of our contribution to this idea of this species. Here's some Cuphophyllus. This is a genus with more decurrent gills and pale coloring. Russocoreaceus is an amazing cedar scented wax cap that has this sweet incensey cedar smell. It's actually named Russocoreaceus, means like Russian leather. Apparently, Russian leather smells like that. But it's one of the best smells I've ever smelled. But anyways, there was a similar species described from Oregon, I believe in the 50s or so, Lawrencii, and it was the type was damaged or lost, and so there was no way for us to sequence it and have see if it was Russocoreaceus or not. But we found a similar species that was like, it looked like Russocoreaceus. Maybe sometimes it smelled, sometimes it didn't, but the sequence came back to be pretty different. And so we thought maybe this could be the Lawrencii that had been described but lost. So it's part of the mysteries being uncovered. Here are the parrot wax caps, Gleophoraceae. You can find this in Wisconsin, like we saw the prairie species, but also up in the forests in the north, they're really abundant. And this is a species complex. These are all these different colors get called Gleophoraceae, but we know that there's a lot of diversity in their morphology, so how they look, their colors, and sizes, their habitats, as well as in their genetic sequences. There's quite a variation, but it's not consistent. So we have these different groups that, as a few years old at this point, this may have evolved for the West Coast, but they not even is all of group 1 that same orangey color and group 3 that pale peachy color. It can actually be multiple colors within each of these groups. So it's pretty confusing and difficult to navigate. But something that I want to mention, I was talking with Sadie earlier about this, is how a project like this kind of helps, you to build on existing species complex. Like the idea of what a species is, what does it look like, where is it found, what does the sequence look like for this? You're able to kind of build on that, maybe break down something that you thought before, like maybe we thought it was restricted to some certain habitat or look a certain way, and we find that actually it can be more variable. Or we find that it might need to have its own new species concept, might need to be something that gets described to science. Um, and so this really helped for us, even though there's still a lot of work that needs to be done to make this reflected in the literature and for us to even really fully understand what's going on. It helps us to kind of better parse apart what we have. So by making these groupings and understanding these complexes, you can actually, and working with people who have different knowledge, you can figure out a lot about a specific group. So I think the Midwest should create an equivalent of this with the wax caps, and probably every group, right? We need all of fungal diversity better documented. But yeah, I'm glad to hear WMS and a few of you guys, Tavis and Brennan and folks have been collecting, and Arnie too, been collecting these mushrooms. But I think it'd be cool to have a concentrated effort on their diversity. Hygrophoraceae phaeococcinea. So this is a European species. There's a very, very similar species found in some grass in a pretty urban area in Washington by an older retired couple. And then an artist, actually Rose Tercy, who you guys may know, who is also involved in WMS. She made this piece of art to showcase like the news of finding this species. And at first we thought, okay, it seemed like a lot of the different features were lining up to be Phaeococcinea, but we ended up not entirely being sure if it was the same or not, if it was a different species or not. There was a lot of debate back and forth. It was a kind of a hot topic, caused some divide within our club. But yeah, it's still ultimately unclear if this is the true representative of the European or not. But it was exciting to find. And then Chromosera. So I have North American Chromosera here because a couple of us were included in this paper that Django Grootmeyers and a bunch of different collaborators worked on for this Chromosera diversity within North America. Because if you looked in your books, everything that kind of looked like this pinkish, purplish gills with a yellow cap was listed as Chromosera cyanophylla, but we knew that there was more species than that. And so they worked really hard to kind of break this down further. I just helped contribute some samples with Sadie, and it was really generous of them to include us in this process. And that's one of the C is the photo of our collection from the mountains in Washington. But so there was, in Eastern North America, there was a Mycena, that was Mycena lilacifolia, and it was ultimately a Chromosera, needed to be moved into that genus properly. And so it is now Chromosera lilacifolia, and found in the East Coast, but also in the Midwest. There's some observations for Wisconsin. And then the West Coast also got their own species. So from Cyanophylla to Lorelea, which is named after Lorelei Norval, who worked with Phaeoclibia and also described some Waxys and worked with Chromosera. But yeah, it was confusing on the east because people were finding these very similar mushrooms, and it was either getting called this Mycena lilacifolia or Chromosera cyanophylla. So it's kind of a mess, and now we finally have a better understanding of the true North American situation. Okay, so here's some of the species that I'm working with for my thesis now and that I'm describing as part of that process. So Hygrophoraceae PNW 18, this is just a provisional code name that we give some of these undescribed species that we make this collection. It doesn't match anything in the books, doesn't have a close sequence match. In this case, the sequence was actually very, very distant from any other species available in like the global GenBank database. And Jean was very curious about this one. And so I was able to make a couple years of collections right kind of smack on the side of this muddy trail in these mixed forests. So a lot of these wax caps end up in North America, throughout North America, being associated with these forests that have some hardwood species, but also some members of the Cupressaceae. So in California, there's the redwood, then you have the cousin, the western red cedar in Washington, and then we have here some other cedars and junipers and such like that. And so a lot of these collections ended up coming from this habitat. And unfortunately, it hasn't been found anywhere else than this one site. I'm hoping to get some more collections of it so that that it can be a better candidate for description. Because ideally, we want to showcase it being found in multiple places. Like it could be so uncommon that it only occurs there, but it just makes a better pitch if it's been found multiple times. We also have a better idea of what the diversity within that species looks like. So if people find it in a different locale than where it's been found the first time, then we can see how consistent or how different the coloration is, the habitat, the size, the spores, etc. And so this one was a bit less slimy the first time I found it, which is in the first photo, but it looked pretty average, like a standard Hygrophoraceae. I didn't really think too much of it, but I collected it. And then the sequence came back to be super out there. It was like branching off all out by itself. And I went back, found it again. These other 3 photos of the slimy one are from the second year, and it was way goopier. It was falling out of my hands and much smaller. So it's kind of interesting to see. This is one that I'm definitely describing and working to get some more collections of that have been made in California. So this PNW 17, that's the provisional code for this species. And these are super tiny. The biggest one I ever found was 3 centimeters, but they're usually sometimes less than a centimeter up to like maybe 2. Super teeny, and they actually, I keep mentioning the side of the trail because a lot of these mushrooms were in very obvious places, but these were actually directly in the middle of a heavily trafficked trail. And that big root, the photo on the bottom right, this large western red cedar root, is you step over it as you're walking on this like muddy hillside near the sound on the campus. And they're just these bright little, they look like little fruits or little cherries, and they're super shiny, they're super slimy, and I just couldn't believe that they were just getting basically stepped over. They got protected a little bit from the overhang of the large root, but that soil is very heavily compacted from people walking on it. And there's a trend with Hygrophoraceae and other wax caps that they can actually thrive in these like more disturbed habitats, like little microclimates like that. And so, yeah, I'm super excited about this find, and it's just a gorgeous little guy that just falls right out of your hands. Here's some more candidates. I'm gonna be working up this PNW 15. This non-prov, this is just like a provisional name that it's not official yet, but this is what I'm hoping to call this species, Hygrophoraceae inversa. And it's kind of because like it's a little bit inverse coloration than what you typically expect for a wax cap. A lot of times the caps are more reddish orange, and then you might get a lighter yellow or a lighter tone on the stipe. And so someone wants to be admitted. If somebody can let them in. It won't let me click. But yeah, so I've actually there's collections of this mushroom throughout a lot of different states, Washington, Oregon, California, and even Montana. Actually, I documented these in northern Montana. So yeah, looking forward to that, and just on the hunt for more specimens right now. Yeah, and then PNW 26, this is one that Sadie found that she's really excited about, and Matt Coons in Washington has also found this one, and it's a summer fruiter, as is the one on the left here too, a lot of times found in the summer. And that might go to show a lot of times people aren't really out looking for mushrooms as much in the summer, at least in the Pacific Northwest. People associate the fall and even the winter with strong mushroom season, but in these wet little like muddy banks and wet areas and logs, there can be a lot of these mushrooms just that people aren't finding and collecting because they're not looking. And so, yeah, this is a pretty unusual one with the brown cap, little scabers. You don't see a lot of brown Hygrophoraceae, but there are some others. And then the provisional name is Flavo-brunea, meaning golden and brown. Some other highlights, not going to go into all these, just kind of showing you some more waxes. You can see how similar they can really look to one another. A lot of these basically just uncovered are either they were going by a European name and then we learned that they are their own entity, or perhaps they're a species complex. And the Irrigatus California 01 down in the middle is one that actually the manuscript describing this species is getting turned in this week, and I helped to describe the species. So it's not going to be called Irrigatus anymore. That's the provisional name. That was a European described species, but we know that there's a couple of North American representatives, and so the West Coast species will have its new name soon, and you'll stay tuned to find out what that is. Yeah, fenestrata, last one I'll mention on the slide, which is cool, is that fenestrate or fenestrated means like windowed. So you can kind of see on the mushroom all the way up on the top, there's this kind of see-through part of the mushroom that kind of shines down into like the stipe, but the cap tissue is actually pretty translucent. So that's why it has that name, and that is also non-profit, needing to be described. And Christian Schwartz has been documenting those for several years, and we're hoping to just pick up that work and get that described. This is a website that is available to check out, Danny's DNA Discoveries, and he is based in Seattle and takes a lot of the Pacific Northwest sequencing results and speculations about those results from various experts and other members, and kind of summarizes them on his website. So you can kind of check what the status is of our thoughts about a species at the time, which is really helpful to be able to just search that on the internet, because sometimes that like this knowledge is had where there's these efforts happening by various individuals, but to be able to like compile it and make it like referenceable is super useful. So I was using this site all the time when I was in Washington. I still use it. I'm really proud they mention us here on the, well, it was originally Hygrophoraceae, now they upped it to the Hygrophoraceae suborder. And they mentioned that if you see under the summary, it says Steve Ness, Lauren Ray, Sadie Hickey, and Eric Chandler are leading a study of waxy caps. It's the South Sound Mushroom Club, not Mycological Society. But yeah, you can check out some more examples of our finds and current standings of our ideas about certain species. And all this is not just for Hygrophoraceae. It's got all you could find any family or genus on this site. See what he says. So for just kind of showing what people are finding, or at least what people are calling these things, it's really hard because of how similar they look, and how I mentioned the situation with the names being applied from other regions, was particularly Europe, makes things really confusing. And so I would take this lightly, but I just kind of wanted to showcase the different observations, the different species that are being observed. This is for all of the USA. Fulvescens, there's a couple other, there's like chlorophanna, there's other yellow things that they all get called fulvescens, is a bright, that golden waxy cap. I won't go through all of them, but yeah, these are ones that you may have encountered or encountered a similar looking thing. The witch's cat hats or witch's caps it's a good Halloween mushroom. I'll talk about my Halloween talk at Funty Fair. But yeah, there's a couple species in that complex. The Cantharella's complex that have the decurrent gills and the scalp margin. Those are also pretty diverse. But yeah, this is what people tend to find throughout the US. And then I did a little grab for Wisconsin as well. It's pretty overlapped, but you'll see a couple of differences, including down at the bottom, there's that Arenia, the brown goblet on wood, but the last one here, the clubfoot Ampeloclytosity clavipes. I actually found this the other day at our club foray, and it is a very funky looking mushroom, I'd say. Sometimes the cap can be really upturned and like kind of really showcasing those decurrent gills, and it really can have a club foot where it's super rounded down at the bottom, but it's less documented, or maybe less common in this region than the West. So get out there and see if you can find it. It doesn't really look very super waxy, but it is in the Hygrophoraceae. I mentioned the West Coast Rare Challenge. I wanted to just kind of showcase which of the species within the 20 species that they have are waxy caps. There are 3 of them. We talked about Flavafolia. which is the yellow one there. And then number 9 is Hygrophoraceae. It's a snowbank, a snow melt associated mushroom species that only grows where the snow line is receding up in the mountains. And it's not super duper rare. There are even more rare hygrophorus, but it's not because it's in that sensitive habitat that it was included in this challenge. Because even though the mountains have these large swaths of forests and different types of what seem like intact habitats, they're very susceptible to climate change because as the temperatures rise, they cannot go anywhere. So if you're a lower land species and things are getting hotter and you need it to be a little cooler, you can move up into the mountains. But if you already live up in the mountains, or if you, especially if you're an alpine species at the tippy top, you don't got anywhere to go. So those are very at risk. And it's a pretty pink mushroom. I gave it its common name. Or they changed it. I guess they call it the rosy snowbank waxy cap. I think I called it the pink porcelain wax cap at one point, and I like that common name. And then Arinia lobata, number 5. That is a weird, funky, like kind of pleurotoid, fan-like Arenia species that grows in mossy wetlands in Arctic and alpine areas. It doesn't really have a stipe. It kind of has like a nubbin. That's why it's called the oysterling. But it has veiny gills. They're not really true gills. But yeah, you could get out west and find some of these things, or maybe who knows? Maybe you'll find them in an unusual region. Okay, features to note for ID. So when you're out, if you think you found a wax cap and you want to try to figure out what it is, I salute you. Take some great pictures and document the substrate. So what was it growing on? Was it on wood? Was it in the soil, in the moss? Keep track of that. That will help to narrow it down, as well as the habitat. If it was a pine forest versus a tamarack versus a sphagnum bog, that will be really helpful as well. And then habit, that just means like, was it singular? Was it clustered? Was it in a fairy ring? Stuff like that. Like, how was that fruiting occurring? And then the viscosity, this is super important. This is something I've mentioned a lot. This talk is the sliminess of these mushrooms. See if the mushroom is slimy or not is the first thing. But if you don't just check the cap or just check the site, check all of the mushroom because sometimes it could be one or the other. And then try to note how slimy it is. So if it's dry, there's really no slime, but sometimes it's like lightly viscid or slimy. It can be like, we call it greasy or sticky, where it's your fingers kind of lingering on it, but it's not goopy. It's just a little bit sticky. And then viscid, we got more like there's a clear slime layer on it that you can see the goo, you could feel the goo. When you pull your finger away, you can see that it's maybe moving with your finger. Maybe not necessarily, but like you can see that there's a slime layer. Whereas glutinous is just absolutely covered in slime. It's just, there's a lot of times there's just slime like globbing off of the side of the mushroom. So that's what we would call glutinous. And then the gill attachment. So a lot of gills on, at least on Hygrophoraceae, are just kind of directly attached to the stipe. They're adnate or adnext, or maybe they're notched. But some of them have decurrent gills like we saw. So you're not going to see free gills. But sometimes you might not see gills at all. So I'll show you a weird one in a second. The cap, yeah, and we saw the weird wrinkly ones too. The cap texture, if there are little scabers or little scales or hairs present on the cap, that will be informative, especially for Hygrophoraceae, or if it's just smooth and glabrous. And then the colors of everything. They're not going to be as definitive as we'd like as a feature, but it's going to really help us to understand the range that given species or complex may have, like with the parrots, right? So document every angle you can, get the gills, the stipe, the cap, a couple different angles, and some scale also would be really helpful in your photos as well. Ideally a little centimeter ruler, or if you need like a penny or something that you can reference. Your hand isn't going to be great because you can't, no one, you can go back and make that measurement from the photo, but other people can't. So yeah. And then the goo. Every time I give this talk, people are like, what is up with the goo? Why are these mushrooms so slimy? And we don't know is the real answer. And there's a couple of ideas. I very commonly, and others that I'm showing here, observe insects and arthropods just like up in the business of the slime. And so in Arnie's photo here, I love this photo of Thylacifolia up north. You see this midge that's just dead and trapped in the slime. And so that could be a deterrent, right? So if you maybe are trying to get in there and either eat some of the mushroom or maybe lay your eggs in the mushroom or something, this could maybe be something that keeps you from damaging that fertile area, because the whole purpose, right, of the mushroom is to make spores and reproduce. So this could potentially be an agent to deter, but that's not proven. It's just more observational that we see insects dead and trapped in the slime. And then Sadie has this cool picture of this parasitoid wasp actually laying its eggs into an insect that's in the mushroom. So a mushroom bypassed the slime, so they're kind of, that breaks that idea down a little bit. But there's a lot of diversity of insects, so maybe some can get through and some can't. One got in, maybe it was a fungus gnat or something, and then this wasp is going to lay her eggs into that insect that's inside of this mushroom. So she found it. They have ways to feel the vibrations if there's wiggling larva or something inside, and they can then use that little bendy ovipositor, this little tube, to lay her eggs in there. So that's just a cool observation. I want to compile these images and share them more with people, just kind of see if we can make a list of diversity of insects that are found with wax cap slime. The other thing that I have observed, at least in temperate areas, is that the lot of wax caps will come out in the winter, and you'll find them in the winter in Washington, Oregon, California. Really, they'll be like the highest diversity will be after the main flush of oh, there's Andrew's in the waiting room, by the way. I don't know why I can't press on it, but yeah, there'll be the kind of the big fruiting of all these different, common fall mushrooms, and then as they start to wind down, we'll see like late November, December, and then even January and beyond, the waxy caps will go full bloom, and we'll see all these different genera and species coming out. And when you're seeing them out, the slimy ones in particular, in contrast with the other mushrooms that don't have the slime, you'll see that the other ones are like cracked. If you've seen mushrooms after they freeze, they just kind of become fractured, and then they end up melting basically after they warm up a little bit, or just like breaking apart in the cold. And not actively growing anymore, and so the slime actually, when you find the mushrooms in the slime, they're like perfectly preserved. They look great. They're embedded in this protective layer, right? And it actually maybe helps potentially to regulate that, maybe the temperature, keep them free from the frost and temperature fluctuation. So that's a hypothesis that needs to be tested. But something to consider is that they also can be slimy in the tropics. So what does that mean? We don't know. You can figure it out. Okay, just a quick highlight, just some microscopic features. This is something you can get into if you're actually curious about identification. I know that there was just a microscope workshop, so unfortunately, the microscopic features on Hygrophoraceae, particularly in like the common genera like Hygrophoraceae, they are not very descriptive. They can help you, and it is necessary if you want to thoroughly document the species to measure the spores and to check them, make sure you don't have something totally different. And they're just going to be kind of classic bean-shaped spores, ellipsoid, kind of like little. This picture of Alan's singer eye shows them well. They kind of got like cute little appendage sticking off from the sides where they're going to germinate, but they're kind of an average spore shape. They're really not super distinctive. They look bumpy in this photo, but they're smooth. They don't have any like projections for the most part, for hygroscopy at least. And then the basidia, the structures that hold the spores, you'll see the mushrooms all the way on the left, and then the structures right to the right of the basidia, little pronged clubs that are going to hold the spores. They can, most of the time people think of them as four spored, but a lot of times in the wax caps, you'll have less than 3 spores, or less than 3, like, they won't even have the little attachment where the spores were. So they'll be like a 1, 2, or 3 spored basidium. This is pretty common. And the true hygroscopy do not have cystidia at all. So these big sterile cells that you may encounter are not present in this genus, but are in other members of Hygrophoraceae. A feature that is important if you are characterizing a wax cap is the pattern of the lamellar trema. And this is just the hyphae, so the mycelial filaments that make up the gill of the mushroom. That's the lamella. That structure of that hyphae is gonna be informative and needs to be characterized. And so you can kind of have this first like spaghetti type. They all look like spaghetti to me, but interwoven where it's like a jumbled, swirly mess. Then they go out, you could see. So this is like the center of the gill, and then the actual the most external part of the gill is where the spores are being made. So you can see those forking basidia off at the end. Then below that one is this parallel type where they're pretty much running, directly against each other, like in a parallel line. And then this other type, figure 3, on the right is a divergent type where they're kind of in the center and then going out towards the edges. I thought it was funny. Michael Kuo on Mushroom Expert says for the microscopy, a Roman aqueduct is required. So this image below showing how you would take a slice of the mushroom, a cross section that includes the entirety of like each part of the gill. So we could see how it's being cut kind of across the mushroom and then really fine to get it really thin. And then you could see the inside of the gill. So he says, Roman aqueduct required, along with a lot of patience and a very sharp razor blade. Good luck. I usually remember some urgent house cleaning or yard work I have to do just when waxy cap sectioning gets ugly. So it can be tricky to get a proper section where you can actually accurately see this arrangement. You have to get a really thin section for the slide, and so it can be frustrating. And so I laughed at that. But we're almost done here. I just wanted to throw in some oddballs for maybe some of the more nerdy folks or, some weird stuff in the family exists. It's already weird enough as it is with like the common genera, but there is a genus called Eonema that's a crust. So there is a crust genus. I think Andrew just joined. Did you know this, Andrew? There's Eonema, the crust Hygrophoraceae. And it's not common, but it exists. And then Hygrophoraceae is a different genus. Oh, that's supposed to say Madagascar. It says Madagascar, it says right now. But yeah, this is a species described from Madagascar, and something that's cool, despite it having this kind of dark coloring, that's cool enough for a wax cap to have a dark colored mushroom. But they actually have stellate spores. They have these like projection spines coming off the spores, which you can see in B in the figure. And that is very unusual for Hygrophoraceae. That's the only member, I believe, that has like ornamented spores. So this is more classic for like the Entomomataceae to have this shape. And then two weird hygroscopies on the right, Hygroscopy roseo pallida, I think. My mouse is covering it, but this is from Panama, and it actually just, you can't really see it super well, but there is actually a veil. If you look closely, you can see there's actually like fibrous veil material covering the gills, which is very, very much not a common feature for a hygroscopy. And then Hygroscopy aphylla. This is a South American gill-less hygroscopy. So like from the top, it looks like it's pretty frilled, but you can see that the cap looks like more of what you would expect, like kind of the color, the texture. You got the different color on the edge of the cap, but underneath it's just a completely smooth, fertile surface. So it is very usual aphylla, like lacking gills, basically, is what that's referring to. Here's some resources. There's not a lot of great resources, and they're kind of scattered in where you can use them and how. Sometimes they're super regional or sometimes they're super old. There's the monograph of North American species of Hygrophorus by Hessler and Smith that described a lot of the species for North America and compiled other existing species at the time. At that point, everything was basically Hygrophorus. So Hygrophorus is the oldest genus, like we saw, is the type genus of the family. And then hygroscopy was created, things were pulled out into hygroscopy, and then things got pulled out of hygroscopy over time. So it is helpful if you want more like detailed, specific information about certain species and to see where they were originally documented from. It is not being printed anymore, but it is not super expensive. I think I got a copy for $8, so if you want to get your hands on one, you probably can. Check out the research by Dr. Jean Lodge. She is one of my committee members and mentors and is just a total wealth of knowledge and powerhouse, still well after retirement. She was the U.S. tropical mycologist based in Puerto Rico for a while and did a lot of documentation of tropical fungi, but also just worldwide wax cap work. She has a big paper from 2014 that has, if you want to like get more nitty gritty with the different sections of the different genera in the family and see how their super big phylogenetic tree breaks things down, I recommend checking that out. It also talks about a bit about the pigments and the ecology. It's like the paper. I think it's on the next slide for my references, but the Mushroom Expert Key is pretty helpful. It's definitely not fully inclusive, but it's a resource. There's an online key for the South Vancouver Island Mycological Society for it's listed as for Hygrophoraceae. That needs to be updated. iNaturalist and Mushroom Observer, you could check out what's been found there. Our Hygrophoraceae, the Lower Puget Sound Project. And then there's some Facebook groups. There's one called Hygroscopy Fungi Forever. That's cute, and people just like post pictures and talk about ID sometimes, but I think it's a European group. And then here's that paper that I mentioned, Molecular Phylogeny, Morphology, Pigment Chemistry, and Ecology in Hygrophoraceae. On the note of the pigments, I know Jane Adams, our pigment focused member and student, is here at the planetarium. And a lot of people I'm sure they ask her the same question, like, oh, this thing is so colorful, like, can I dye fiber with it? And in the case of the Waxies is no, you can't just you can't gather that something is going to transfer, that the color of something bright will transfer to fabric. It's all about the actual chemistry and how that's going to bind to the material that you want to dye. So unfortunately, I have not heard of anyone successfully dyeing any fibers with wax caps, but if you end up figuring that out, do let us know. It actually, the cool thing with the pigments, just one more thing, is that they're actually betalain pigments. So they're the same pigments that beets have and members of the Caryophyllae, some cacti with like bright red coloration. Yeah, so if you want to read more about that, check out this paper. You can read more about their endophytic lifestyle in this paper here and kind of the conservation importance, why these wax caps have been so useful for them to be able to conserve land in the UK. And then some other sites here that you can check out. Yeah. This is just, I wasn't sure I was going to include this at all, but this is from that big paper. So if you want to really get nerdy with it, this is from 2014, so it's even been updated. Like there's new things we've learned since, but this is like ideally showing us how all these different genera are related in this family. It's not as scary as it could be with all these nice colors, so maybe you'll feel like trying to dive deeper into it and we can talk. Yeah, and then, okay, also the Continental Mycoblitz for Mycota Labs is happening starting tomorrow. So if you are finding cool mushrooms, did somebody say something? No? Is that Mariah? I don't know. Anyways, yeah, you can collect mushrooms, document them on iNaturalist, go to Mycota Labs website and or mycoblitz.org apparently, and you can get your mushrooms sequenced. And they're suggesting now a contribution of $3 per specimen, but they will not turn anyone away who cannot pay for their samples, or if you want to do a dollar donation, you can basically go out during this time period, October 17th, 26th. You can document any mushroom, doesn't have to be a wax cap, but there are still wax caps fruiting. So I will encourage you to document those and collect those and get those sequenced. And then you'll dehydrate them less than 100 degrees, ideally, or less than 120 or so, and save them. And
I
then
think there's going to be some really good references also on the Mycota Labs website, probably about their exact specifications for how they want you to collect.
yeah, I'm over time. So anyways, that's it. Yay!
We do have one question. We don't
Sure.
probably have a lot of time for a ton of questions, though anyone can stick around as long as they like. But the first one is just, does the gillless waxy cap have any waxy parts to it? Like, is it still waxy?
I kind of find that despite the gills are definitely like the waxiest part, but I find that the caps are kind of waxy, especially when they've gotten like a little bit wet. They kind of have like some of them have a thicker cap tissue, and I feel like it kind of has a waxy feeling. So based on the pictures, I've never seen that species, but I kind of feel like it might be a little bit waxy on the feel of the cap, but I can't say for sure. And maybe that smooth, maybe that like there are basidia and spores on that surface, that smooth surface underneath. Maybe if you scrape some up, maybe it would feel waxy. I don't know.
Someone report back.
Maybe it's just like kind of an analogy of it is like it's like a couple of gills that just got stretched out flat and glued down on the underside. So maybe they share that same texture. Well, maybe you should go to Ecuador and find out and let me know. I'll go with you.
Is there anybody who would like to unmute themselves and ask a question aloud? You can go for it if you want, or put it in the chat.
I was curious if the American grasslands have the same relationships, if there's been a lot of hygroscopy found in American grasslands too, or if it's mostly observed in the UK.
There have been observations throughout North America. A lot of the times they're concentrated on the west. There's been some researchers in Oregon who have focused on prairie habitats, so they've really mostly included the prairies there. I would say it's like underrepresented and understudied in the other regions, and still even in the West Coast. But they have been documented throughout North America in grasslands. But the type of grasslands are going to vary between the continents too, and within each one there'll be variation. So it's like that low plant diversity in the British wax cap grasslands. Like at least it's my understanding. I'm not a botanist, but like I associate the prairies here with a high plant diversity. I know someone could maybe add into that, but there's definitely more diversity for sure. And so it's not the most comparable. So that might be why we're not seeing those same trends. But people are still finding them in these grasslands, sometimes in grassy lawns that are more that even do have chemicals put down on them that are like mowed and managed versus like more wild grassland areas. So they're found in both in this continent, but not, I think people aren't looking to as much as they need to. We know that they're not. So if people can get out, I would encourage you to document, because like, Jamie found the parrots today. And so a lot of people think we're past peak mushrooms. Maybe we are past peak diversity, but there's still interesting things occurring. So yeah, but Biddy Roy, if you want to check out some of her studies for the North American prairies and the fungal relationships there, yeah, her work will include the North American
Could you
prairies.
type her name in the chat?
Yeah, she's an ecologist. She always says she's not a mycologist, but she is. She's doing some incredible work with like tons of sites throughout North America. Yeah, I gotta look more into.
If I recall some of the work on the British grassland waxy caps, they actually argued for mowing of the grasslands to help preserve them. Is that correct?
Yeah, I think that it's like tricky because one of the papers I was looking at recently was saying how like it's suspected that partial grazing, not constant grazing, but there is meant to be a cycle of getting the grass down lower, because the mushrooms like they are like going to be out. The grasses grow very tall, so they're going to be it's going to be hard for them to maybe get out and get their spores out. So they do rely on some of that grass coming down, but also not constantly, and not like a pouring in of nutrients. So I think that makes sense that would be recommended because it would be helpful, but you don't want like a constant mowing situation happening.
We do have a question in the chat from Haley and Neil, which is with a lot of specimens looking similar, what are some things we can do to narrow down if we should keep them, like for future sequencing and collections, or should we just keep them all? Like, are there any priorities that you would say? Like, we've kind of identified like North American grassland waxies and similar, but
yeah, I mean, I personally say keep all of them, because even if we think we have, like, if you open your field guide and you say, oh, it's Hygroscope fulvescens or it's Hygroscope coccinea or Cantharellus, there's still a lot to resolve with these species complexes. It's actually becoming more and more, as we sequence more mushrooms, we're learning that we know less about them than we thought, and that we had less of a grip on the range for some of these species and how many species are going under one name. So I would say if you have the time and energy to preserve all the wax caps you find. Don't then do it. I would say do it. If you want to really narrow it down, it's going to take a little bit of time talking to some folks who are more clued in to like what we need more of. Like I can recommend collecting in prairies, but in terms of specific species to collect, I mean, the Chromosera work just came out, so now we have an idea for those. Gleophorus, the parrots and stuff, collect those. Hygrophoraceae need so much work. I mean, there's really no going wrong with it because that's part of how we ended up making some of these discoveries is we were seeing things and we thought like, oh, this is something we see all the time. But then the day we decided to document it and send it in, we were glad because they came back to be informative. So I would say collect as many as you're capable of. If you find a lot, sometimes in Wisconsin or different regions, I'll find like dozens of one hygroscopy. You don't need to like physically collect all of the mushrooms in the patch. Leave some for sure. But ideally, if you can get a range of sizes included, so like ideally more than one is best for a collection. If you can get like 3, 4, 5, 6 mushrooms, that'd be great. And especially if that's going to be described one day, having that range is going to be really helpful and getting photos with scale with those different sizes so that we can go back and actually get those measurements for like a paper. So take photos, take good photos, different angles. They're so beautiful. It's great because they're a fun mushroom to find and photograph and document. So that really helps the cause, you know. Max says, I
Very
know some interesting forking gills on Kupophyllus species Tavis showed me on his property. Are there any other species that might have this trait? From what I gathered, they tend to be straight. Forking. Yeah, I've seen some forking, but it's been a while. I wouldn't say it's this like limited to Kupophyllus. It might have been an environmental thing. If anyone wants to add on about their experiences, or Sadie's, I know there, if you want to add something. I don't see a lot of forking, but yeah, Kupophyllus are where they have really thick flesh and those decurrent gills, so I wouldn't be surprised if they would like anastomose and like combine sometimes, but I haven't seen that too much myself. Something I do see a lot, which I haven't really seen any information about in literature, is like the double forking, like the stipe being doubled, and like there's like looks like the identical stipe is like made with a second one and they're connected. I think Mariah was suggesting like maybe it's like a good structural support thing to have like, double up, and instead of making a whole new mushroom, put the energy into like bulking up your stipe to hold up your cap if you've got these big thick gills. But I see that throughout a lot of genera, but I don't see forking as much in the gills. That's cool though. I'd like to see photos. Any other questions? I was like just going off about so many things. Hopefully it was coherent. Any comments? Was it informative?
Yeah,
I'm gonna say a thumbs up.
All
Great,
right.
great job, Laura. There was a awful lot, but on the other hand, I learned a great deal too. So
Cool.
thank you so much.
Yeah, of course. Yeah, it's a big scope. But yeah, someone's asking, don't waxy caps, aren't they eaten in Europe? And yes, I forgot to mention that. There aren't any toxic wax caps that I'm aware of besides the few that have the psychoactive components and those lichens. But people do eat Hygrophoraceae and Kupophyllus and Hygrophoraceae. Hygrophoraceae are more commonly eaten in Europe than they are here. I only know like one person who eats them here. They're not toxic, but they're not like super flavorful, so they're kind of the edible but not incredible category. And then Hygrophorus are really some people like eating those out in the West because they're like thick and meaty and will get the flavors that from what you cook, but they don't have a lot of flavor on their own.
We highlighted in the WMS newsletter collecting for eating Kupophyllus virginiana or something in that complex that we have around here. And I think that
Yeah,
that one
some
is
people will eat those.
apparently rated as very good, but it is also rated as like a know what you're doing. This is a small white mushroom with gills. Know how to ID it. But then once you do, maybe try it.
Totally. Yeah, I view it as like survival mushrooms, kind of. That's in that category for me. I haven't heard anyone be like, they're so delicious. The
No,
flavor.
I mean, the people I haven't yet tried myself. I think a decent quantity of Cuphophyllus virginianus, but it was reviewed as really, really good. So, by the people who had. Many people are saying in the chat that they loved your presentation, and
Aw.
I just wanted to say thank you. So you make sure you saw that, and wanted to say
That's very nice. Thanks everybody. I'm really grateful to be here.
thanks, Lauren.
And yeah, I'll see some of you at the Fungi Fair. I'll be presenting on Halloween mushrooms and fungal folklore, so come check that out. And I'll also be vending some fun items.
Yeah, and Fungi Fair is October 26th, coming up. So October 26th, Sunday, this month.
Yeah. Come talk to me about wax caps.
yeah.
I'm definitely not, I should have disclaimed, I'm not an expert. I'm really mostly an enthusiast. I'm working with some of them, but still I don't feel that makes me an expert. Just it's helping me learn a lot, but there's so much I don't know about this group. So if you want to like just talk and speculate with me, I'm happy to, or correct me. Sometimes I mix things up, but I'm happy to talk to you about waxies anytime.
Thank you so much. Yes, there will be a foray at the Fungi Fair, a little short one, just on the grounds there at the Bavarian Beer House, where we're going to hold it in Glendale, Wisconsin. So with that, I'll say good night, and we'll see you later. Bye.